Hi friends, so today we will learn Amphibian Nerve Muscle Experiment, which is a fixed 5-mark question for your practicals. Actually, this is a computer assisted learning as we are not doing this with animals. So let's get started. These are the list of graphs that you should study in this particular topic. Total we have 8 graphs. First one is simple muscle twitch, then the effect of various strength of stimuli on simple muscle twitch, effect of changes in temperature on SMT, effect of multiple successive stimuli on muscle contraction, study of fatigue in the skeletal muscle, velocity of nerve conduction, effect of load on muscle, and at last the measurement of isometric contractions using nerve muscle preparations. This is a model question. This is how you will get question from this part for 5 marks. So first you have to identify the graphs and you have to answer for the below sub question. So as you can see here, the first question is to identify the graph and comment, that is for 1 mark. Define latent period and list the causes for the latent period. Write difference between isometric and isotonic contraction. So this is how you will get a question. Before discussing the graphs, we should see how the experiments are done and what is the need of doing this. So as this is an amphibian experiment, we are taking a frog here. Why we are taking a frog? Because as we know, frog is a cold-blooded animal. So the preparation will survive more than the warm-blooded animal. Then we'll see the steps for the nerve muscle preparation. So first step, we have to take a frog and we have to pit the frog. So what do you mean by pitting of the frog? Pitting of the frog or pitting is a procedure where we are distracting the brain and spinal cord of the frog. So the sensations like pain will not be felt on that frog, but all other organs and muscles will be intact. So that is the first step. So after pitting, we have to cut the skin on the legs. Then we have to remove the skin. Then we have to expose the nerve and muscle of the frog. Here we are taking gastrocnemius muscle and sciatic nerve because these are the longest nerve and the largest muscle. So this is the gastrocnemius muscle and this is the sciatic nerve that we exposed. After exposing the nerve and muscle, we have to connect the nerve and muscle to the electrode, then to a kymograph. So if there is any kind of movement which is happening to the muscle, means this will be traced on this moving kymograph. Now we'll see a video. So after seeing this video, you will understand the procedure more precisely.
Place a pithed frog on a dissection board with its posterior surface facing upwards. Cut open the skin of the frog using a pair of scissors. And remove the skin from the abdomen down the lower extremities. Cut the thigh muscles with the scalpel to expose the sciatic nerve. Pin the muscles down on either side. Then, use a seeker to free the nerve from surrounding tissue. Insert a pair of forceps under the nerve. Moisten the nerve and the muscle with a few drops of Rigor's solution. Finally, fix the muscle at the knee with a pin to stabilize its position. Apply 6 volts to stimulate the nerve. Observe any changes of the calf muscle.
So these are the steps. First step, pitting the frog, then applying amphibian solution to the frog and nerve in 5 minutes of interval. Why we are using a solution here? Because it makes the preparation intact. Why particularly Rigor solution? Because it contains inorganic salts and concentration which is very similar to our extracellular fluid. So it will help to prolong the life of our preparation. Then we have to cut the skin around the leg and remove it. Then isolating the gastrocnemius muscle and sciatic nerve, connecting it to the electrode and giving the threshold stimuli. Then we will get a graph. Now we'll see important question from this part. Why we are taking frog for this experiment? So as we discussed before, frog is a cold-blooded animal. So the nerve muscle preparation survival rate is more compared to that of the warm-blooded animal. Then it's easy availability, easy to handle, less expensive, and it is easy to dissect. Therefore, because of all this reason, we are taking frog for this amphibian experiment. Next, usually for nerve muscle experiment, the sciatic nerve and gastrocnemius muscle of the frog are used. Why? Because gastrocnemius muscle is the largest muscle and sciatic nerve is the longest nerve in the frog. Also, this nerve and muscle are very easy to locate and dissect. And gastrocnemius is a bigger muscle, so on contraction, it will produce more force to lift the liver and good magnitude of contraction. Then gastrocnemius muscle cannot be fatigued easily. Now we'll discuss the first graph, that is simple muscle twitch. This is a normal graph that you will get by this normal experimental setup. And we have to study each point and each component of this normal graph. Then only you will be able to understand the remaining graph. So now we'll start. So this is a line and this is the baseline. This is a point where you are giving the stimulus. So this is point of stimulus. Now this is the point where the contraction is started. So this is point of contraction. Now comes the graph. So from here the contraction is starting till here the contraction will be there. So this period is called the contraction phase or contraction period. Now comes the relaxation phase or the relaxation period. Then comes physiological curve. Physiological curve is nothing but it is coming because of the inertia of the liver. And from here till here we are getting the contraction, and this is the point where we are getting the maximum contraction. So this point is called PMC, that is point of maximum contraction. So this is the contraction period, relaxation period, physiological curve and point of stimulus. So there is a time gap or time interval between the point of stimulus and the contraction phase. Okay, and that is called the latent period. That is called the latent period. Physiological reasons or the causes of latent period we will discuss later. So these are the component of a normal simple muscle twitch. Another important thing in this graph is these small curves. These are the time traces. So from this time traces, we can able to calculate the time interval of each phases. So as you can see here, so this one curve is around 0.01 seconds. So in this contraction period, we can see 4 graphs, 1, 2, 3, 4 curves. So contraction period is having 0.04 second of duration. Relaxation period is having 5 curve. So relaxation period is 0.05 seconds. And latent period is 0.01 seconds. So that is the importance of the time traces. If you are drawing this graph, don't forget to draw this time traces. Okay. So time traces, the point of stimulus, the latent period, contraction period, relaxation period, physiological curve and point of maximum contraction. So these are the important points that you should learn in this simple muscle twitch. And this is how you will get the graph for exams in a black and white chart. So this is the simple muscle twitch graph and these are the important points. PS is the point of stimulus, LP is the latent period, CP is the contraction period, RP is the relaxation period, PMC is the point of maximum contraction and physiological curve. That is nothing. It is coming because of the inertia of the liver. We'll see the important question from this part. Define latent period and what are the causes, and which is the very important question. So the latent period is a point from the point of the stimulus to the point of the onset of contraction. Its normal duration is around 0.01 second or 10 milliseconds. And what are the causes of the latent periods? So we know for the nerve conduction, so first it should conduct along the nerve, then it should reach the neuromuscular junction, then it should cross the neuromuscular junction, then there should be excitation, contraction, coupling. So for all this, it is taking some time. So that is the latent period. Then it will take time to overcome the viscous resistance of the muscle and the time to overcome the inertia of the recording lever. So because of all this, there is a gap between these. So that is the latent period and this question is very important. What is physiological curve? So this is nothing, it is coming after the SMT and it is just because of the inertia of the liver. What should be the speed of the drum while recording the graph? It should be at 640 millimeter per seconds. What are the difference between isometric and isotonic contraction? Give example. This is another important question from this part. So Isotonic and isometric contraction. Iso means what? Same. Tonic means tone. Metric means like measure or the length. Okay. So isotonic contraction means the tone will be constant here. The length is changing. Isometric means length will be constant or same here and tension or tone is changing here. Okay, so that is the main difference. So we will see. So isotonic contraction is a contraction where the muscle shorten in length but the tension remains constant. Then external work is done here. Not much heat is produced. Then it requires much sliding over of the filament. Coming to the phases, the latent period, contraction period and relaxation period are of shorter duration. Examples are jogging, when we are doing exercises, lifting weight, all these are the example for isotonic contraction. Coming to isometric contraction, this is a contraction where muscle length is not affected, it is constant, but tension in the muscle is changing. No external work is actually done here. Greater amount of heat is produced because of that. Then it does not require much sliding over of the filament. Contraction period, latent period and the relaxation period will be longer. Examples are pushing a wall, then action of anti-gravity muscle when a person is standing. These are the examples for the isometric contraction. Now we'll study the second graph, that is effect of temperature. So how we are going to do this experiment? So that is with Rigor solution. So if we are studying the warm effect, we are giving warm Rigor solution. If we are studying cold temperature, we are giving cold Rigor solution. So this is the baseline and this is where we are giving point of stimulus and this is the normal graph. This is the point where we are getting the contraction. Then contraction period, relaxation period and physiological curve. This is the normal graph. And now this is the graph where we are giving the warm Rigor solution. So what is happening here? This is the point of contraction of the warm Rigor solution. So actually we are getting the contraction faster. Coming to the force of contraction, the force of contraction is also higher. So we are getting more force of contraction and we are getting the contraction earlier. Now we'll see the cold effect. So this is the point of stimulus and this is the point where contraction started for cold Rigor solution. And coming to this force of contraction, the force of contraction is reducing. So that is the effect of temperature. As you can see here, this is a normal graph where the Rigor solution temperature is 26 degrees Celsius. This is warm Rigor solution around 38 degrees Celsius. This is cold Rigor solution around 16 degrees Celsius. This is the point of stimulus. We are giving stimulus on the same point for all these experiments. So this is the first graph, latent period. This is the warm. So the latent period is reducing here. This is for cold. Latent period is increasing here. Coming to the contraction, this is a normal graph. This is the warm. So force of contraction is increasing in warm and force of contraction is decreasing in cold Rigor solution. So that is the effect of temperature. Now we'll see, this is the graph. So what is happening in a cold Rigor solution or a cold saline? So the duration of contraction period, latent and relaxation period is increasing, decreasing the height or the force of contraction. In warm Rigor solution or warm saline, duration of the latent period, contraction and relaxation period is decreasing and it will be increasing the force of contraction. So that is the effect of temperature on the nerve muscle preparation. Coming to the important questions, how warm solution increases the force of contraction? So it will increase the force of contraction by increasing the conduction velocity, increasing the rate of neuromuscular transmission, increasing myosin ATP's activity, decreasing viscosity of the sarcoplasm and decreasing the inertia of the liver. How cold solution decreasing the force of contraction? It is by decreasing the conduction velocity, decreasing the rate of neuromuscular transmission, decreasing myosin ATP's activity, by increasing the viscosity of the sarcoplasm, overcoming the inertia become slower in the cold solution. So when we are doing the experiment, the effect of the warm Rigor solution or the study of the warm Rigor solution should be taken first or should be studied first before the cold Rigor solution. Why? Because cold Rigor solution, once we are used, it will be inactivating the muscle. Once inactivated, it is very difficult to revive it or it is an irreversible phenomenon. So when we are doing this temperature effect, first we should study about the warm Rigor solution, then only we have to do the cold Rigor solution. What is the temperature of the Rigor solution beyond which there will be damage to nerve muscle preparation? So the temperature should not exceed around 42 degrees Celsius. Then what will happen if we increase the temperature? So the proteins in this muscle will be getting denaturated. So it should not be exceed 42 degrees Celsius. Now, why is it important to use maximal stimulus while recording the graph? So here for the experiment, we are giving the maximum stimuli to get the maximum contraction, to get the maximum response. So we should ensure that all the motor unit is activated simultaneously for the maximum response. What is the effect of the temperature on activity of the muscle in human? So there comes the importance of the experiment. So in humans during exercises and all, this is coming to action. So the efficiency of the skeletal muscle contraction during experiment is increasing because metabolic activity is increasing, so the temperature will be more, so the efficiency of the skeletal muscle contraction will also be more. So that is the effect of temperature on human muscles. Now, is the effect of temperature is a violation of all or none law? Exactly, it is a violation of all or none law. The all or none law is valid only in a normal environment condition, like normal temperature, normal pH and all. So this is an expectation, or this is a violation of the all or none law. Now, what is heat rigor? So rigor is a familiar term for us. Rigor means stiffness or the state of contraction of the muscle. So this rigor is happening because of the heat. So when the Rigor solution's temperature exceeds more than 45 degrees Celsius, what is happening? The muscle protein will be getting denaturated. Once it is denaturated, actin, myosin filaments will not be able to carry out their functions. So coagulation of muscle protein will lead to stiffness and shortening of the muscle fiber, and this is an irreversible phenomenon. That means the muscle remains in a state of permanent contraction. So the rigor which is happening because of the heat, and that is called heat rigor. What is cold rigor? So the rigor or stiffness or permanent contraction of the muscle is happening because of cold temperature. So on exposure to severe cold, if it is reduced more than 0 degrees Celsius or below, what is happening? The tissue and the muscles become frozen and stiff, and the excitability of that muscle fiber will be lost, and the muscle will remain in a contracted state. But this is not permanent, this is a reversible phenomenon. So if we are giving a warm environment or warm temperature or warm solution to this muscle fiber, it will be back into the normal state. So this cold rigor is reversible phenomenon and heat rigor is irreversible phenomenon. Now one more rigor we all are familiar with is rigor mortis. Mortis means death. So this rigor is happening after some hours of death. So after the death, all the body muscles will be stiff and it will be rigid and short. So that is because of the depletion of ATP. So if the body is in a contracted state, crow's braids will be formed with the myosin head and actin filament. So to detach or to release this crow's braids formation, there will be ATP. After death, there will not be availability of the ATP. So this crow's braids of the myosin and actin will be continued and muscle will be in a rigid state. After some hours, the cellular lysosome will come into action. The cellular lysosome will release particular enzymes. These enzymes will distract our muscle. The rigidity will be gone. And this rigor mortis is very useful for the forensic expert to fix the time of the death. Now the third graph, effect of two successive stimuli on muscle contraction. So here we are giving two successive stimuli of same strength. And this stimuli we are giving is during the different period of the graph. Here first we are giving stimuli during the latent period of the contraction. So what is happening, we'll see. So this is the point where we are giving the stimuli. Okay, this is the first stimuli point, and this is the point where we are getting contraction. Then we have contraction period, then relaxation period, and point of maximum contraction. Now we are going to give another stimuli during this latent period. So here we are giving next stimuli, that is PS2. What is the effect or what is the response? So there is no response at all. Okay, there is no response because the latent period is falling in the absolute refractory period. Okay, what do you mean by absolute refractory period? It is a period if we are giving any kind of stimuli. If we are giving maximum stimuli also, there will not be any response. So that is absolute refractory period. So we will not get any response if we are giving stimuli during the latent period. Next, we are giving second stimuli during the contraction phase. So what will happen, we'll see. So this is the first point of stimuli. This is the first point of contraction. So we are giving stimuli, contraction is started. During this contraction period, we are giving another stimuli. So this is the point of second stimuli. What is happening here? So here during the contraction phase, we are giving second stimuli. So the second contraction or the contraction here is summed up. So this is called summation. What is the causes? What is the reason behind this? So here this is already in a contraction period. So calcium, the released calcium level is very high. So if we are giving again a stimulus, means there will be more and more release of the calcium ion. So this contraction will be summed up, add up together, and we will get a contraction with more force. So that is the effect when we are giving stimuli during the contraction period. This phenomenon is called summation. This is called wave summation, or it is the temporal summation. Now we are giving stimuli during relaxation period. So this is the point of first stimuli. We are getting contraction. So during this relaxation period, so this is coming to relaxation. During the relaxation period, we are giving the second stimuli. So what is happening? So this will be superimposed. What is happening? During the relaxation period, most of the calcium will be taken up, but still calcium ions will be there. So if we are giving second stimuli, this calcium ion will be taken up, and the second contraction is superimposed during the relaxation phase of the first one. So we will get a contraction again with more force. So that is called superposition. So here the second contraction is superimposed over the relaxation period of the first one. So that is called the superposition or superimposition. Okay. Now are giving stimuli after the relaxation period. So this is the point of stimuli, first point of stimuli, first point of contraction. So once all over is done, we are giving again a second stimuli. So what is happening? We will be getting again one contraction, relaxation. Again force of contraction will be high. That is because of a phenomenon called beneficial effect. So the second contraction is getting benefited because of this first contraction. So what is the mechanism of beneficial effect? So it is a phenomenon in which second stimuli is getting benefited by changes which is produced during the first stimuli or during the first contraction. The reasons are, so during the first contraction there will be release of more calcium from the sarcoplasmic reticulum, there will be increase in temperature, there will be decrease in the internal viscosity, and there will be decrease in the inertia of the recording system. So all these benefits, second stimuli or a second response is taking up all these benefits, and there will be increasing the force of contraction. That is called beneficial effect. Okay. Now, why is maximum stimulus is used in the experiment? Again, it is to ensure that all motor units are simultaneously activated. Then only we will get proper response. Now, the fourth graph is effect of increasing strength of stimuli. So here we are giving multiple stimulus, that is with increasing the strength. So first we are giving sub-threshold stimuli. So what is threshold? So threshold is a minimum stimuli where we will be getting a minimum response. Sub-threshold is less than the threshold. So if we are giving sub-threshold means we will not get any kind of response because it is not in a threshold stimuli. It is sub-threshold, it is minimum threshold. Okay, then if we are giving threshold, we are getting response, that is also minimum. So if we are giving threshold, we are getting minimum response. Then if we are giving maximum threshold, we are getting maximum response. If we are giving more and more stimuli threshold, that is supramaximal threshold, we are getting maximum response that is of same as that of the maximum threshold. So here the response in the maximum and supra-maximum is same. Why? Because of odd or none low. So either it will not produce any kind of response or it will get maximum response. Okay. So in sub-threshold or sub-minimum threshold, we are not getting any response. Why? Because no motor units are recruited here. Why? Because we are giving minimum or sub-threshold here. Okay, now we are giving threshold, so we will be getting minimum contraction because minimum number of motor units are recruited. Then we are giving maximum stimulus, so we will be getting maximum response because maximum number of motor units are recruited. Then supra-maximum stimuli we are giving and we are getting maximum response same as that of this one because already all motor units are recruited in the maximum stimulus. So there is no leftover motor units to recruited here. So we will be getting the same response, same maximum response. Okay. Now one question, what is motor unit? So it is the functional unit of the muscle contraction in the intact body and it consists of a single motor neuron and all muscle fiber innervated by it. And what is Heineman's principle? Heineman's size principle. So here this states that strength of the contraction is increasing gradually. So if it is increasing gradually, first small motor units will be recruited. Then only the large motor units are recruited. And if additional motor units are recruited means more tension will be generated. So this is a motor unit. So first all small motor unit will be recruited. Then large motor units will be recruited. So first size first, then all. So this is in an ascending order. Then if additional motor units are still recruited means we'll be getting more and more tension. So this is the end of this class. So we'll continue with part 2 of this nerve muscle amphibian graph experiments. Thank you.